Hi, and welcome to the 17th Florida Keys Mosquito Control District Oxitec Public Educational Webinar. Today, we're going to talk about the 2022 project, the project that's going on now, and what this means at launch. With us, we have Chad Huff. Chad Huff is the Public Education and Information Officer of the Florida Keys Mosquito Control District. We have Rajiv Vaidyanathan, the Director of U.S. Programs at Oxytec. And I'm Meredith Bensom, a Florida native and Head of Public Affairs at Oxytec. So, we've been hosting this series of public educational webinars to share information with residents of the Florida Keys and to provide forums to answer questions. These webinars are open to everyone. They are recorded and made available along with their materials after the event. All questions relating to the webinar topics will be answered. We may group similar questions together. And if we run out of time, we will accept questions in writing via florida@oxytec.com. You all can start to submit your questions now. You don't have to wait until the Q&A part starts, but we will give about 30 minutes for the question and answer. If you can leave your questions in the chat box, they will be anonymous. Members of the audience will not see who is submitting the questions. And if you are a Keys resident, please let us know which key you live on, and we will give priority to Keys residents. We want to answer your questions first. So our agenda, we'll spend the first half hour talking about this year's project and at launch, and then we'll go through the Q&A. And Chad, now over to you.
All right, Meredith, thank you. Just kind of a refresher on how we got here. Dengue fever is an ongoing challenge here in the Florida Keys. It was only 2 years ago, we were in the midst of a pretty hefty dengue outbreak. Most of those cases were confined to the Key Largo and Tavernier area, and but 72 cases was pretty substantial, and it was at least it was about a decade before that we had our last outbreak. So it was a pretty substantial outbreak, and it had been quite a while since we had it. Of course, because of the climate in the Keys and because of our proximity to water and the way the population is centered and the type of demographics and the transient population we have here, the potential for mosquito-borne disease such as Zika, chikungunya, yellow fever, heartworm, and other diseases is always a concern, and we're always under kind of a heightened awareness for that. As in a lot of places in the country, mosquitoes, the Aedes aegypti mosquito in particular, in the Florida Keys demonstrates an amazing resistance to pyrethroids. This is one of the pesticides, one of the few approved pesticides that the district can use to adulticide. And we just returned from a conference 3 or 4 days ago where this is a growing problem nationwide. It's not just in the Florida Keys. It's with mosquito controls across the country, and we all use the same type of adulticide products, and the Aedes aegypti is showing a remarkable resistance to this nationwide. And then besides from being resistant, the Aedes aegypti provides mosquito control a lot of other challenges, not the least of which is the fact that it breeds and chooses to live almost exclusively around human beings. It wants to be around people. Therefore, it breeds within a few feet of homes and residences and businesses where people are. And it breeds in very cryptic breeding sites. It only needs a small amount of water in any type of container. So it's very adept at living underneath decks and and being in bird baths and that sort of thing. So it's something that mosquito control has to deal with because it takes a while to find those cryptic sites or to educate people about them. And mosquito control, Florida Keys mosquito control, among them, we're always looking for additional tools. There just aren't that many that are available to us. And so any new tools that we can put into our toolbox, it's worth our time to investigate that and to show our due diligence in that regard. And that's what we're doing here. Of course, for environmental impact, this particular product checks that box. There it does not, has not been, at least in the year that we have tried it out or that we've been using it, there has not been in detrimental environmental impact. And in fact, we'll see that again this season. And 1 year of working with the product, we've determined that we're dealing with a non-biting male insect. And in fact, of the insects that we released last year, the Oxitec mosquitoes, they were all, as advertised, non-biting males. And that makes it very attractive to look at, because it just doesn't have the impact on anything other than other mosquitoes. And that's what we're interested in. So all those things considered, it makes it a good product for us to look at. And as long as we follow the guidelines, which we have been doing with our Oxitec partners, and we look forward to doing that again this season. Rajiv, I think this one's yours.
Thanks, Chad. I'll talk a little bit about our technology, and then I'll also reiterate some of the results, kind of go into that in a little more detail. So indeed, the reason that we're working with Aedes aegypti is because it is an invasive species, it's an important vector of viruses, and because it poses a challenge to traditional mosquito control technologies. One of the benefits of using a genetic approach to controlling Aedes aegypti is that you are targeting one species. Some people say, but what about the mosquito? As if the mosquito is a general entity. There's no such thing, right? There are over 3,000 species of mosquitoes, and there are plenty of native mosquitoes to Florida. Aedes aegypti is an invasive species. It is not native to North America, South America, or Asia. And so by using male mosquitoes, male Aedes aegypti, to mate with the local invasive female Aedes aegypti, you are targeting the one invasive species that is also a competent vector of a number of viruses. In addition, because we are releasing exclusively males, and we'll go into some of those results now, this is safe, non-toxic, non-allergenic, and again, very targeted against one species, and we're using only males. We've had experience now in Brazil for a few years and in the Florida Keys for 1 year now. And one of our goals this year is really to see how well we can suppress some of the local populations of Aedes aegypti. When we say male only, people ask, well, why are you making such a big deal out of saying male only? And that's because only female mosquitoes bite and feed on blood. Male mosquitoes do not have mouth parts, they don't have the saliva, they don't even have the gut machinery to feed on or digest blood. Male mosquitoes cannot feed on blood. And that's why we use them, because the males mate with the local female mosquitoes, resulting in fertilization. The female mosquitoes lay eggs, and of those eggs, all their male larvae survive and all their female larvae die. And we'll talk about what that really means. And when we say traceable in the field, what that translates to is that our male mosquitoes have two genes. One is that female specific gene, and the other is a fluorescent protein. And so our larvae are indistinguishable from any other Aedes aegypti larvae. When you look at them, you cannot tell the difference. However, if you look at them under a red filter, under a microscope, ours fluoresce red like a Christmas tree. And so we can get a full number of how many mosquito larvae we're collecting. And of that, we have a numerator, right? We have a subset of how many are fluorescent. So we have an idea about the penetration into the local mosquito population. So let's talk about what that really means from last year. Our self-limiting gene in female Aedes aegypti larvae killed 100% of the females in which that gene was expressed. And that's remarkable, right? And we're really, really thrilled about that. So as I mentioned, we collected thousands and thousands of mosquito larvae, of Aedes aegypti eggs, hatched them out. Of those, over 20,000 were fluorescent. We took each of those, put them in separate cups, allowed them to complete their metamorphosis, go through a pupa stage, and emerge as an adult. Every single fluorescent larva emerged as an adult male. Now, those are not statistically improbable. That is entirely because of the genetic mechanism that kills the female mosquito larvae. So it was 100% effective. Any female that got the gene from her father, right, essentially from one of our males, died as a larva. And so we, one, released only males, and two, when our males mated with local females, among their progeny, all the female larvae died, male larvae lived and emerged as adult males that could then go on and mate. We tested different dose rates because we had to see what would be operationally feasible and useful. One of our goals of the project is to really provide this as a tool to FKMCD and other mosquito control districts. And anyone from a mosquito control district would ask, okay, at what rate should we be releasing this? So we tested it at two different rates, and we'll be testing at a rate this year as well. When we say our Oxytec males performed well, what that really means is how far did they fly? How well did they mate, right? Those are two fundamental metrics of success. Well, we know that our males from the release boxes that you can see here in this slide flew approximately 50 meters or about 150 to 200 feet. That means that their behavior is identical to male Aedes aegypti that are non-Oxitec. Aedes aegypti are always kind of starving. They do not fly far, and they're always energetically starved. The fact is that both male and female Aedes aegypti rarely fly very far from human habitations. Female Aedes aegypti feed and breed in and around people's homes, and male Aedes aegypti go where the female Aedes aegypti are found. So one, we know that our males flew about 50 meters from the release sites. Two, we know that they mated with local female Aedes aegypti. And three, we know that among the larvae that came from those eggs, only males survived to adulthood and all females died. We also know that kind of unexpected, because we didn't plan on testing necessarily, but we're very happy with the serendipitous data, that we put out black cups in which we collected Aedes aegypti eggs. And those are the ones we brought in to really do the majority of our quantification. But in addition, we also collected eggs and larvae from other what are called oviposition or egg-laying sites. That means jet skis, bird baths, the kind of places that Chad talked about, buckets, wheelbarrows, children's toys. So we collected Aedes aegypti larvae from a lot of other sites in people's backyards. We brought them in, and we found fluorescent larvae in those collections as well. What that tells us is when the local invasive female aegypti females mate with our males, their egg-laying behavior is unaltered. That means that they can deliver to people's backyards where traditional mosquito control sprays, essentially, cannot reach. It also means that where they lay their eggs, those cryptic sites, as Chad mentioned, that we cannot see, we don't even know where they are, And if you don't know where they are, you cannot target them. The females lay their eggs in those cryptic sites. So this is operationally really good news, because it means when we release male mosquitoes and they mate with female mosquitoes, those mated female mosquitoes go off and they lay eggs in sites that we cannot see and do not know exist. Finally, the most important, maybe out of all of this, is that we did not release a single female mosquito. We set up our boxes that you see here in people's backyards, where they have very graciously volunteered. And by the way, thank you to their eagerness and their support throughout the Keys. It's been wonderful, and we're looking forward to this year as well. So we set up those boxes, and then we set up a cohort of those boxes. Same water, same eggs, same food, but inside a mesh cage. Just like the butterfly cages the kids use when they use insects in school. From those cages, we vacuumed Monday, Wednesday, and Friday, opened them up, and we looked at every single adult mosquito that our release boxes released. Every single adult was a male. And so we have two discrete lines of evidence. One, when we collect eggs and larvae and we detect fluorescence, every time those mature to an adult, it's a male. And every single adult that we collected from our release boxes was male. We have 100% of male emergence, no females at all. And now a word about our 2022 project. We work closely with the US EPA, the Environmental Protection Agency. And what we're doing in 2022 is a regulatory pilot. This is not an operational deployment. We're not working with Florida Keys Mosquito Control District to actually suppress mosquitoes. This is part of a regulatory pilot. And we want to be able to show that at a certain dose rate, for a certain duration, this can actually be a feasible operational tool. And some of this might go into some detail, so we'll walk through it. Our aims for 2022 are to show that, yes, we can reiterate our data from 2021, that they're all males, that they fly about 50 meters from the boxes, that they mate with the local invasive female, Aedes aegypti. But in addition, we also want to collect data to push this forward in terms of commercial registration. So that way, this is a tool for mosquito control districts and also for consumers. In order to do that, the EPA granted us an extension of an experimental use permit, or an EUP, that was granted in 2020 for the Florida Keys, and what we used to actually do the work in 2021. For 2022, we have received their EUP that will enable us to repeat this process until 2023. So that makes sense, right? It's essentially a one-year extension. In order to grant us this EUP extension, the EPA reviewed and responded to public comments over a 30-day period that they allowed public comments to be made. And all of their risk assessments, along with the approved field protocol, are available at regulations.gov. So all of their testing, all of their evaluation, all of their expert external evaluations, and all of our reports are available publicly on the EPA site. That's step one. Step two is on a state level. So the Florida Department of Agriculture and Consumer Services, or FDACS, approved an extension of the permit that was granted in 2020. It is now feasible until 2023. Therefore, last year, we started our deployment of boxes in April 2021, And we ended in November 2021. This year, we've started in the first week of May. So the boxes are out. And by this time, third week of May, our male mosquitoes are out in the Florida Keys. So 2022 has started. We're very excited. Thanks to the EPA and to FDACS, we've started our 2022 deployment of male Aedes aegypti. What does that mean in terms of the regulatory process? As your Tinder profile might indicate, it's complicated, but this is how we break it down. We first applied for and received a federal permit, and that is all the scientific literature and the external third-party expert review of what's called risk or hazard. After the federal EUP, we then submitted and got the evaluation from the state, and that's FDACS. We then worked with the Florida Keys Mosquito Control District, the FKMCD, and with their board and with their staff so that they knew where we were going and what we were doing. And so they're part of that project planning. And with the approval of FKMCD, we could set up our surveillance and our box releases. And then we loop it all back, all these data, where we collected, what we collected, the numbers, the significance. We loop back and submit those results to the EPA. So it was that dossier, it was that collection of data that we resubmitted to EPA, which enabled us to get a permit for this year. How do we collect? Well, think about butterflies, right? Butterflies have 4 parts to their life cycle: eggs, larvae or caterpillars, a chrysalis, and an adult butterfly. Might be hard to believe, but mosquitoes have a similar life cycle. They have an egg, an aquatic larva, an aquatic pupa, and then from that pupa emerges an adult mosquito. We collect 2 of those life stages. We collect eggs and we collect adults. And we collect eggs, as you can see, with these small plastic cups. By putting these cups out with a paddle, female Aedes aegypti are attracted to the smell of the stale water, frankly, and they lay their eggs on those paddles. We bring those paddles in, put them in clean water, hatch them out, and now we can count the total number of eggs, count the total number of larvae, and count that subset of fluorescent larvae. So it gives a measure of not only the Aedes aegypti force in an area, but also what percentage of those mosquitoes are the progeny of one of our males. In parallel and in addition, we also collect adult mosquitoes. The concept is pretty simple, and there are a lot of different traps that are set up like this. Essentially, it's a pie pan with a light underneath and a fan, and we hook that up to a battery. Underneath the fan is a mesh bag. The mosquitoes are attracted to the light, they're sucked down by the fan, and they end up in the bag. We then bring those mesh bags into the lab, freeze them, and then put them under the microscope, where we get to step 3, monitoring for adults and adult mosquitoes. So we might use these microscopes to identify species of mosquitoes and to count the number of Aedes aegypti, especially the Aedes aegypti that are ours. In addition, if you look at the picture at the bottom right, you'll see that the top half of that are larvae. They're hard to tell because it's a off-white larva. You can't really tell the difference between one of ours and the locally invasive species. When you look at it under a red filter, that red fluorescent protein shows how we can count. 1, so we get a number, and 2, how we can distinguish the fluorescent larvae from the non local invasive Aedes aegypti. What are we doing in 2022. It's a big project, and it's a little more expanded than last year, even though we're looking for many of the same measures. All male, how far do they fly, how well do they mate with local females, all of that is true. In addition, we're doing two kinds of pilot studies that will tell us some really important stories for the EPA. So we can take this, hopefully, for commercialization. The first we call Project B. It's a small neighborhood study. So essentially, we are going to look at a full neighborhood where we have a good distribution of release boxes. So this is a strategy that whole neighborhoods or HOAs can use to understand what is the release rate, what does saturation mean. It's not going to tell us anything about flight radius, right? Because you can't really distinguish if the adult male that you collect is from this release box or from that release box. So it's not really going to tell us about flight radius, but it will tell us about suppression. So this is a really important step to understanding what does release mean in terms of controlling a local pest population. Baby steps. The next big study we call Project D, and that's a household study, whereas Project B is in Bravo, is really to give us an idea of what kind of a release, what kind of a distribution is good for an entire neighborhood or an entire HOA. Project D, as in David, is a household study. If you could purchase these boxes and do a release in your backyard, what does that look like? How many boxes do you need? What is the release rate? What proportion of the Aedes aegypti around your home are actually affected? And how long does it work for? So this is a lot of trapping. The number of boxes and the geographic distribution is limited in Project D. It's expansive in Project B. But in Project D, the trapping and surveillance is extensive. So we really want to know, if you set this up in your backyard, how far are they going to fly? How well do they mate with the local female Aedes aegypti? And what effect does that have on the subsequent population? Those are the two big projects for 2022, neighborhood-wide and small household scale. The third isn't really about homes. It asks a very simple question, but it's a hard measure. And that is, how far do the male Aedes aegypti fly when you just release them? We're collecting at 50 meters. How far can that go and for how long? So in this one, for Project E, what we call Mark, Release, Recapture, or MRR, the male mosquitoes will be covered with basically chalk dust. We use a powder, and then we release them, and you're going to hear stories about pink mosquitoes or blue mosquitoes, and that's because of the powder. And then we trap them at 50, 100, 200 meters, and that is really answering one question: how far can they fly? And that is important for operational needs. So you kind of bring Project D and Project E together. Leave Project B out of it for a second. Project D tells us how well do they cover a small household. Project E tells us how far they can actually fly. And by knowing those two, we can really tailor it for the consumer, for the backyard, for the homeowner. Project B, as in Bravo, was started the first week of May. As I mentioned, almost all of our male mosquitoes from the release box were found about 50 meters or 150 to 200 feet from their release locations. And we've designed that to achieve mosquito coverage throughout a neighborhood. Again, it doesn't really tell us how far they fly, but it does tell us how well we get coverage. So that's a lot. That's a lot to digest. I'm going to pause and hand it over to Chad to talk about, to move from kind of the operational to the community level support.
Thanks, Rajiv. Very complicated, but you explain it very well. Thank you very much. Very, very nice job. It's a good point here to remind of folks that that we have a wonderful independent advisory group that we meet with quarterly and share with them some of the results that we've seen, and also talk over with them about particular issues, whether it's outreach or procedurally or anything like that. And they're a wonderful, diverse group, and we need to give them credit here for participating with us. First is Dr. Jorge Ray with the University of Florida Entomology Lab, Mr. Bobby Eady, who is a wonderful partner here at the local health department, Dr. Doug Mader, a very respected veterinarian from here in Marathon, who is just a wonderful consultant with us on issues pertaining to our animal friends, heartworm, that sort of thing. And George Fernandez, who is our partner from the Butterfly Conservatory in Key West, a wonderful partner. And all four of these folks have been great resources for us. And we are very much look forward to continuing to work with them this season. I also would like to give a nod to Meredith and Oxitec that we have kept a pretty robust public engagement, public involvement program running during what we would call our off season during the fall and winter. This season, a lot of due to COVID concerns, a lot of events were kind of pushed back, and we have really been heavy on events in the March, April, and May timeframe. And Oxitec has been with me at all of those events, and it's been very nice. We participated at a lot of high-profile big events and talked to a lot of people, and it's great to get their feedback and to chat with them about the project and let them know what sort of potential there is for us here in mosquito control. And we look forward to continuing that robust public involvement program through now and through the upcoming mosquito season, which really has just started to really roll in earnest here in the last couple of weeks. So we look forward to continuing this transparency and this public involvement. And I forgot who I was tossing to here. I'm sorry.
I can take this, if that's okay.
Yes.
Yeah. This is kind of reiterating, but also expanding about what we're doing this year. So I hope that it's clear. We have regulatory approval from FDACS, right? And we've started Project B. That's the neighborhood-wide distribution. So that means prior, in April, we started surveillance, right? We want a good baseline so we understand how many eggs are we collecting, how many adults are we collecting before we release any males. So we've already started in April our pre-release surveillance in the Project B sites and in the control sites. And just remember, when we say control, that means there is no treatment, right? So this is no male releases at all, but we're looking at what the populations are in the treated and untreated, namely control sites. Wonderful news to share. We visit homes in these neighborhoods. We ask people, would you like to be a part of this project? And what does be a part of this project really mean? You can help us collect eggs, collect adults, or would you like to host a release box? And wonderful to say, we are, just like last year, we are oversubscribed for the trap hosts and for the release box hosts. In other words, we have more people signed up to participate in this project than we actually need. And that's wonderful. The support from the communities in the Keys has been overwhelming. It's been eager, it's been gracious. And we thank the residents of the Keys for just being so welcoming to us. We've started Project B, Project D and E. So that's the homeowner and that's the mark release recapture. They'll follow later in the mosquito season when there are a lot more mosquitoes. It doesn't make sense to do them now. And I'll toss back to Meredith. And I've also gotten a message here. Please remind everyone to put their questions in the Q&A box.
Thanks very much, Rajiv. And everyone, we'll turn to the questions now. And please don't be shy. No one's going to see what your name is. If you're curious about something, this is a great time to ask, and we'll answer your question. So our first question, Rajiv, I'm going to toss to you. I hope you know the answer, maybe just a rough estimate. You might not know exactly, but the question is, how many mosquito traps do you have out this year?
See there. So when we say trap, right, a trap could be an egg trap, and those are the black cups with water in them, or they might be a fan trap. So that's an adult trap. So first of all, there are different kinds. The cup collects eggs. The fan collects adults. Okay. Of the sites, we have about 30 right now. 30 of the fan traps. So those are 10 traps per site. So that would be 30 for the experimental, 30 for the control site. So about 60 adult traps. You can do the math for the eggs. That'll be a little more tricky, but at each of the sites where we're doing releases, we have 35 egg cups. So that's about 210 of the egg cups and 60 of the adult traps. Probably more detailed than you wanted. But we're looking at different metrics, right? We're collecting the eggs, bringing those in. We're also collecting the adults.
Great. Thanks, Rajiv. The next question is for you too, and it's about how many of these male mosquitoes come out of each box.
That will depend on the rate, and so we are looking at different rates of release. Typically about 1,000 per box per week. And the final answer would depend on how many weeks they're out. So it's very important that we don't confuse what the predicted is with the actual. And that's why when we put out a box, we also put out a cohort box in a mesh bag and then actually collect all of those. So at the end of this year, we'll have an actual number, right? Right now, it's a predicted number, how many we expect to release from each box and for how many weeks. And at the end of this year, we'll compare that and say, these are the number of boxes. This is the number per box, and this is the duration. So we will actually calculate how many we released, just as we did for last year.
Great. Thanks, Rajiv. The next question, Chad, why don't you start answering it? And I mean, we might weigh in from the Oxytec side too, but the question is the public support this year stronger or weaker than last year?
Yeah, that's a good question. I don't know. I would definitely characterize from my own personal perspective, this year, public support has been stronger. but that doesn't mean I want to categorize it last year as weaker. Let me explain. We've had a lot more events because of COVID, uh, and so I have talked to, and we have with our Oxitec cohorts at these events. As a virtue of having many more events that have come online, we have subsequently talked to a lot more people, and certainly of all the people that we talked to, by and large, they've been very supportive, and I've been surprised that they usually bring the subject up. And what I mean by that is when they approach our event, or they approach, I'm wearing a Florida Keys Mosquito Control District shirt, and we're in front of a Florida Keys Mosquito Control District tablecloth, and so they come and they talk about whatever mosquito, or it could even be a bug issue, and then a lot of times on their behest, they will bring up, oh, by the way, how is that project going? And I can always turn and I can say, Rajiv is here to, he will talk with you about it, and we'll talk together about it. So I've been blown away with how much more support I feel like that we have this year, how much more awareness we have of the project. But again, I guess I take kind of a politician's answer there. I certainly feel like it is stronger, but that doesn't mean that I thought last year was weak, if that makes any sense. But it's a good question. I take a few phone calls a week too, where people are just curious about it, and have heard something about it and want to know more about it. And those calls are up as well. In comparison to once we were in mid-project last year. So good question, but yeah, I would definitely say we've had a lot of strong support this year.
If I may, so that's the observation, right? And I agree with Chad. I think the support has been uniformly positive. It's been very good this year. I'm interested in the mechanisms underlying that observation, and having now knocked on probably, I don't know, 6,000 doors or more, and spoken to so many Keys residents, the number one reason Florida residents are interested in hosting a box or participating in this project is they want newer, better tools for mosquito control. I mean, it's almost simple to say that, but it's the number one response. Because I ask people, so why do you want to participate? They say, oh, anything to reduce the number of mosquitoes. So they're really supportive. The second most common reason people join us is because of a good, positive relationship or experience with the FKMCD. It is ubiquitous. Oh yeah, my neighbor works for FKMCD. My cousin works for FKMCD. So Chad and Andrea, we can do this because of you. We can do this because FKMCD is so trusted and has such a good name that when we come in and say, hi, we're from Oxytec, we're partnering with the Florida Keys Mosquito Control District. We would love to collect mosquitoes on your backyard. We'd love to release male mosquitoes in your backyard. People say, absolutely. And that's because of the relationship with FKMCD. And the third most common mechanism underlying people's response to participate in this project is a personal experience with dengue or chikungunya or Zika. When people are aware that, yes, mosquitoes transmit a disease, or if they themselves have been personally affected by it, they are eager to sign up. So everything Chad said is true. And I think that those are the three mechanisms underlying it: newer, better tools, good existing relationship with FKMCD, and an awareness of the role that mosquitoes play in transmitting viral diseases.
Thank you both. This next question will go to you, Rajiv. When will this pilot finish, and will the data be published alongside last year's data? If so, when?
It'll be so a couple questions there. Yes, it'll be the duration of this year. Essentially, we do have two main projects: Project B and Project D. Project B is neighborhood wide. Project D is a small household, and so we want to hit it during the time of worst mosquito problems. So we do expect the duration of actual releases will correspond with the summer and the worst time for Aedes aegypti in the Florida Keys. Now, having said that, right, last year we had to stop and restart twice. 4th of July weekend was Tropical Storm Elsa. And then the second week of August was Tropical Storm Fred. And that pushed us back. We were delayed by months last year because we had to bring everything in and then we had to put everything out. So let me caution by saying our plan is to continue the releases through the summer, cease at the end of summer, and then no more releases, but we will be collecting eggs. So we have a good idea of the duration of the effect, i.e., how long does fluorescence and the self-limiting gene actually exist in a population. So we'll have to do that. The caveat there is if tropical storms occur, it could well delay us. Now, the second part of your question is about publication. Yes, the reason we are replicating the project this year is because more replicates provide greater statistical robustness to any experimental design. And so we will compound the data from 2021, our results from 2022, and our very first obligation, our very first need is to submit those data to the EPA, right? So it has to go for EPA and FDAC's review. At that time, we can start writing the manuscript and submitting that to a journal. So that's not going to happen until next year. We have to compound all that data, bring it together with last year, submit it to EPA and FDACs, and at that point, we'll have what we need to actually put together a manuscript and submit it for publication.
Thanks very much, Rajiv. Chad, the next question is for you. And the question is, when was the last dengue outbreak in the Keys?
Dengue was the last time we had and we need to quantify that, locally originated is what's the concern. That is where a person apparently contracts dengue on the premises or in the area. And the last case was the outbreak that we had, not last summer, but summer of 2020, where it was 72 cases and they were all confined to the Key Largo area. That was the last locally originated case that we have confirmed. Of course, that information comes from the health department, but that was the last one we were advised of was in 2020. But we are always on alert for it. It's something that because as I talked about earlier, because of where we are in the world and our proximity to other islands in the Caribbean and the transitory nature of the population, and the fact that dengue is much more prevalent in other parts of the world, and that it can be brought in at any time and then passed on to a local mosquito population. It's something that we are always concerned about. So I'm saying 2020, but I am very loudly knocking on wood here at my desk, hoping that we don't hear of one soon. But that was the last time.
Thanks very much, Chad. Okay, sorry. 1 second to bring up our next question. Oh, I'll answer this one. It says, do you need more homeowners to sign up? We'd be happy to subscribe if you do. The short answer is yes. please sign up. We've made it really easy for you. You can go to our project website, keysmosquitoproject.com, and you'll see little tabs at the top. One of them is Join the Project. You can go there and you can sign up to either host a box or a trap or join our project listserv. There are hundreds of Keys residents, and really now people from all over the nation and the world signed up to that listserv, and we send updates out fairly regularly. But you can sign up there. And just because we say that, well, we're oversubscribed and we have wait lists, doesn't mean don't sign up, because it's true, we have wait lists throughout the Keys. But, Rajiv talked about the design of our different projects. Like B is running right now, and we haven't started running D and E. And we do not know exactly where those projects are going to be. So, it may be, because this has been happening throughout, that we have a lot of people signed up in a neighborhood that we're not going to. And then maybe there's a neighborhood we are planning to go to, and we have signups, but maybe we don't have everyone that we need yet. So please do sign up if you're interested. We want to know, and you may hear from us. Okay, next question. This is a little bit of a different one, but that's okay. I'll take this one. Why not? The question is, without giving away financial details, does the Oxitec business model still look promising? And I'll just say yes. And if there are any follow-up questions to that, we're happy to answer them. But yes, the Oxitec business model still looks promising. And some of you watching may not know that we work not only with mosquitoes for public health, but also self-limiting insects for food sustainability. So we work with agricultural crop pests, and there's really a lot of need for these technologies that are effective and environmentally sustainable. So we're proud to be working on those. Next question, Rajiv, I think you're a great person to take this, because you are spending time in both places. The question is, how is this pilot similar or different to the one that will happen in California?
Okay. They're very similar. Our basic metrics have not changed. We still want to ensure that all the adult mosquitoes that emerge from boxes are male. So that hasn't changed. That's true for Florida and California. We want to ensure that the flight radius is comparable. So that hasn't changed. We want to ensure that they're mating with local females, and we'll be measuring the number of fluorescent larvae as a ratio of all the larvae. So all of those metrics are the same. Fundamentally, the big difference between Florida and California is that one of the important metrics for Florida is actually controlling or reducing our baseline numbers of invasive Aedes aegypti. We were unable to do that last year because we were not collecting a whole lot of local invasive Aedes aegypti. So if you're collecting 20 females per trap, and then you're collecting 10, is that really a 50% reduction? Well, no. That's just variable in a population. So it was hard to make that comparison between our B sites and our control sites last year. This year, there are a lot more Aedes aegypti. It was a wet spring, and we're seeing higher numbers in our control sites. And we've also changed our B sites to areas with historically higher numbers. So the most important distinction between the Florida and California projects, all other things being equal, males, flight radius, mating success, all that's the same. But what's different is that in Florida, we will be quantifying what the change is in the local invasive pest population.
Thanks very much, Rajiv. This next question, I'll take this question. The question is, if this is the success you are all hoping for, when would these male mosquitoes be made available by the EPA? And I think that question is asking, when would we have our approval from the EPA to sell our mosquitoes commercially in the United States? And the answer is, if all goes well, that would probably be 2024 or possibly beyond, but we don't think it would be before 2024. And I should note, we just had our commercial launch of our mosquitoes in Brazil the end of last year. So if you are living in the state of Sao Paulo, you can order our mosquitoes in little box with their food and have one of these delivered to your doorstep and do this yourself at home. So that is our reality in Brazil today, and we look forward to that reality in the United States one day. Okay, next question. Rajiv, this is good for you. How long do the Oxitec male mosquitoes live for? And is that the same as invasive mosquitoes?
Yeah, the metrics, what are called life table metrics for Oxitec mosquitoes, are equivalent to those of the invasive population. So the males live for a few days, just like the males, just like the wild type males. Mating success is on parity, on the same rate as the invasive males. So the important point, and that's where numbers come in, is that we essentially overflood, right? So we have to kind of outcompete the local male mosquitoes by sheer number, because our males are as competitive in mating, and our males live as long as the invasive males, and so we have to give them a numbers advantage. So we release a higher ratio so that they can mate with the local females. But all the other metrics are the same.
Great, thanks, Rajiv. The next question, either one of you can answer, maybe both of you want to. And the question is, can all mosquitoes carry diseases?
Chad, can I take this? It's like, that's my thing, man.
I know no other expert that would be better equipped to answer this, but I could give the brief answer, but I think you should give the longer one.
Why don't you start?
No, it's not the only disease that only species that carries disease, but in the Florida Keys, it is by far and large the biggest one to worry about, because she vectors so many diseases. She is an equal opportunity distributor of several different diseases.
So Meredith, would you mind repeating the question? Because I want to make sure that we understood it.
Sure. The question is, can all mosquitoes carry diseases?
There you go. The answer is no.
The answer is no.
The answer is no.
But that's one of the biggest ones, is with the point I was trying to make.
Yeah. So there are 3,500 species of mosquitoes. Take a second to think about that. Okay, 3,500 species of mosquitoes, and they are as different from one another as, you know, a dog is from a cat. They're very, very different. Now, among all of those, only a handful are really important as vectors of diseases. Among the Aedes aegypti and Aedes albopictus are the most important competent vectors of dengue, chikungunya, Zika, and yellow fever. Malaria has completely different vectors in the genus Anopheles. West Nile virus is transmitted by mosquitoes in the genus Culex. but not all mosquitoes can transmit. And that is some very fundamental research. There are only a few members of the rogues gallery that can actually imbibe an infectious blood meal, have the virus or parasite replicate in their gut, and then transmit upon feeding. Aedes aegypti is an exquisite vector for two reasons. One, it is a competent vector for a number of different viruses. And two, female Aedes aegypti feed multiple times during ovarian development. So every other mosquito species, the female feeds, develops her eggs, lays her eggs, and then feeds again. And that cycle could take 3, 4, 5, 6, 7 days. Aedes aegypti females are not gulpers, they're sippers. And that's important because that means they can feed multiple times from host to host to host, and the time in between is abridged. Aedes aegypti is one of the most important mosquito vectors of human disease on the planet.
Thanks very much, Rajiv and Chad. And we're a little bit past time, so I think we'll wrap up now, but really appreciate all the great questions and everyone's interest. If you do think of questions later, please send them to florida@oxytec.com. We're very responsive there, and the video and the slides from tonight's webinar will be made available on our project website in the coming days. All the past webinars are there too. And we just really thank everybody for participating. We're continuing this webinar series, so please join us next month and beyond. Thank you again. Thanks, everybody.
Thank you.
Thanks, Chad. Thanks, Meredith.